Alright, so from next segment, what is the continuing burden of communicable disease? Well, communicable disease can cause epidemics, which are outbreaks that spread rapidly, above, say, a normal expected levels, and then pandemics, which are a global spread. And they can also become endemic, meaning they're regularly present in the population, causing ongoing illness and death. In the mid 20th century, efforts in medicine and public health provided a temporary relief. Medical advancements in treatment and public health initiatives aimed at preventing or eradicating infections seemed to give hope that communicable diseases might eventually become a thing of the past. But this turned out to be a temporary lull. By the late 20th century, we saw then the emergence of new diseases and the reemergence of previously controlled diseases. For instance, HIV and AIDS, recognized in the early 1980s, has since killed over 50 million people worldwide. Other diseases like Lyme disease and the West Nile virus weren't recognized in the US until the late 20th century, but have since spread. In addition, mosquito-borne illnesses like Zika and malaria pose a growing risk of spreading or returning to the US. In addition to the large number of epidemic communicable diseases, there are those that can cause a pandemic. A pandemic is an epidemic that occurs globally or in a large area, crossing international boundaries. A recent example is COVID-19, caused by the SARS-CoV-2 virus. Now, the greatest pandemic danger is the ability of RNA viruses to mutate rapidly and cross over to humans, often from animals like bats. Zoonotic diseases, diseases that jump from animals to humans, can spread quickly, especially when we interact with wildlife or destroy their habitats. Some RNA viruses that pose a potential threat to humanity, and may sound familiar, include other coronaviruses, influenza, HIV, polio, Ebola, Zika, measles, and many others. Right. So now how do we establish that a potential pathogen is the etiology of a communicable disease? Traditionally, Koch's postulates were used to establish the cause of communicable diseases, particularly for bacterial infections. These postulates were designed by Robert Koch, who discovered the bacterium responsible for tuberculosis. Koch's postulates state that to definitively establish a cause and effect relationship between a pathogen and a disease, four conditions must be met. First, the microorganism must be found in all cases of disease. Next, the microorganism must be isolated from the host and grown in pure culture. Third, the microorganism must be the cause of disease when introduced into a healthy host. And four, the microorganism must be re-isolated from the experimentally infected host and shown to be identical to the original pathogen. However, these postulates were too rigid for some modern diseases, especially viral infections, and this is where the pathogen can't always be isolated in pure culture or is not as easily grown in the laboratory. To address these limitations, a set of modern Koch's postulates was developed by the National Institute of Allergy and Infectious Disease to better account for viral and other emerging pathogens. These modern postulates consider advances in molecular biology, genetic testing, and other scientific methods that can help establish the etiology of disease, even when Koch's original postulates cannot be fully applied. Right, so now here, these are the four original postulates developed by Robert Koch to establish a causal relationship between a pathogen and disease. So the first condition is that the pathogen must be found in every case of the disease. Second condition is the pathogen must be isolated from the disease host and grown in pure culture, which means it is free from other microorganisms. Third, a healthy host must develop the disease when the isolated pathogen is introduced, proving the pathogen caused the disease. And then fourth, after the disease is reproduced, the same pathogen must be recovered from the newly infected host, showing that the same microorganism is involved. Now, oddly enough, tuberculosis, the disease that Koch himself studied, could not be shown to fully meet these postulates in every case, despite being the disease for which the postulates were originally developed. So, of course, this highlights how challenging it can be to apply the postulates universally. All right, so now fast forward to what we call the modern Koch's postulates. The modern Koch's postulates have been updated to account for challenges with viruses and other pathogens that cannot be cultured easily or don't always meet Koch's original criteria. And these postulates now focus on the three key criteria. First off, we have association, there must be an individual association between the presence of the pathogen and the presence of the disease in human beings. This means that the pathogen must be consistently found in those who have the disease. Second up, we have isolation, meaning the pathogen must be isolated either through traditional culturing techniques or by using genetic sequencing to detect the pathogen's DNA in those with the disease. The pathogen should be found in most, if not all, of the affected individuals. Then third, there's transmission. The pathogen must be able to be transmitted to another individual, and the same disease must develop in the new host. This confirms that the pathogen is the contributory cause of the disease. So these three steps, association, isolation, and transmission, are the foundation of modern Koch's postulates and allow us to establish a causal relationship between pathogens and diseases, even in cases where Koch's original postulates might not apply. All right, so as we touched on a little bit of transmission on the last slide, we'll be going more in depth on the potential routes. So there are a wide range of possible routes of transmission for communicable diseases, depending on the pathogen. And these routes include person to person, and is the most common route, where diseases spread directly from one individual to another. Examples include respiratory droplets, like with the flu COVID-19, or through direct contact, like with skin infections or STDs. Then we have animal species to humans, which is known as a zoonotic transmission, where diseases are spread from animals to humans. Common examples include rabies, HIV, since it is believed to have originated from primates, or the Zika virus, which are transmitted by mosquitoes. Then it can be a physical environment to humans, is another route. Some diseases can be transmitted via the environment, so this can include contaminated water or food, such as cholera, or contaminated surfaces or airborne particles, like in the case of tuberculosis or Legionnaire's disease. Understanding these routes of transmission helps in creating effective prevention strategies to stop the spread of diseases. Public health measures such as vaccination, sanitation, quarantine, and vector control, such as mosquito nets, can help reduce the transmission of many diseases. All right, now what we have here is table 7.3, which outlines the major methods of transmission of human disease and provides examples of each method. The table helps categorize how diseases are spread, including the routes like person to person, animal to human, and environmental transmission. Now it's important to know that some diseases such as HIV or hepatitis B have more than one potential route of transmission. For example, HIV can be transmitted through sexual contact, blood, or from mother to child during childbirth. Understanding these different methods is crucial for developing targeted prevention strategies and public health measures. All right, so now what measurements help us predict the extent and the speed of transmission? So to start off, we have what is called R0, which is a measure of how many new infections an infected individual will cause in a population of people who have no immunity. So basically a completely susceptible population. This number is used to estimate how communicable a disease is and the potential for that disease to spread widely. If R0 is greater than 1, the disease is capable of spreading exponentially because each infected person will then infect more than one person, leading to potential epidemic or even a pandemic. For example, diseases like measles and COVID-19, which spread through respiratory droplets, tend to have a higher R0. For these diseases, R0 can be much greater than 1, indicating rapid transmission. R0 is also helpful in estimating the level of immunity, so either protected either through vaccination or infection, is needed in the population to prevent an epidemic, which is known as herd immunity. So the calculation for the level of herd immunity is, you say 1 minus 1 divided by the R0. So for example, if R0 equals 1.5, around 33% of the population needs to be immune to prevent the spread. If R0 is 2.5, that needs 60% immunity. R0 is 4, then about 75% of the population needs immunity. Now, if R0 equals 10, then about 90% of the population needs to be protected to stop the epidemic. And COVID-19 was thought to have an R0 above 10, meaning a large portion of the population needs to be protected through either vaccination or natural immunity to control the spread effectively. Okay. Now, touching on the subject, so what else matters besides R naught in determining the course of an epidemic or pandemic? Now, R naught is still an important measure of disease potential for spread, but it doesn't address the speed of transmission or the effectiveness of control measures. Several other factors can also significantly affect the course of an epidemic or pandemic, such as one of which is what we call the serial interval, or SI. Serial interval refers to the time between an individual's exposure to the pathogen and the development of a transmissible disease. For example, for COVID-19, the serial interval was typically 2 to 4 days, which led to its rapid spread because individuals became infected very quickly after exposure. Now, when you combine R naught with serial interval, we can predict how quickly a disease will spread. For example, for COVID-19, with an R naught of 10 and an SI of 3.5 days, the number of new cases could potentially increase 20-fold in just 1 week. Now, next up, we have the period of communicability. The period of communicability is a time frame during which an individual can transmit the disease to others. For COVID-19, it was found that people could transmit the virus for about 5 days after the onset of symptoms or after a positive test, as long as the symptoms were resolved. But in some diseases, people can continue to transmit the pathogen even after recovery, and these individuals are called chronic carriers. So these examples can include diseases such as HIV, hepatitis B, and hepatitis C. Now, a major challenge in controlling disease like COVID-19 is asymptomatic transmission, meaning individuals who don't show symptoms can still spread the disease to others. So in the case of COVID-19, it was found that at least half the transmissions occurred from the individuals who would even have symptoms at the time of transmission. Asymptomatic transmission makes it more difficult to control the disease because we don't know who is capable of transmitting it. This complicates the efforts to reduce R naught to 1 or less, which is needed to stop the epidemic. So now, what tools are available to public health to address communicable disease? There is a range of tools available to public health officials to address the threat of communicable diseases. These tools are designed to prevent, control, and manage the spread of disease, and we will take a look individually in the next couple slides. But some of the ones you can see here, we have a barrier protections, screening and contact tracing, immunization, antibiotic and antiviral treatment, and elimination or eradication. All right, so now for this slide, we'll be going in depth on one of the tools mentioned previously, the barrier. Barriers against communicable disease can range from very simple interventions to more complex measures. For example, something as simple as hand washing is a basic barrier that can help stop the spread of many diseases. Another example could be an insecticide-impregnated bed nets, which are nets that hang above bed and repel and kill mosquitoes. That could be another example. One of the most effective barrier protections for preventing, in this case, a sexually transmitted disease is the use of condoms. They are widely considered to be the best way to prevent the transmission of many STDs, including HIV and gonorrhea. Masks have been a major tool in controlling the spread of respiratory diseases. During the COVID-19 pandemic, 3-layer surgical masks, and to a lesser extent, cloth masks, were shown to be partially effective in reducing the transmission of the virus. 5-layer masks like the N95 are found to provide excellent protection, especially in high-risk settings. Next up, we have isolation, which involves separating individuals who show symptoms of the disease from those who don't. And this helps prevent the spread of infectious agents from symptomatic individuals to healthy people. Then there's quarantine, which is used to separate and restrict the movement of individuals who have been exposed to a disease, even if they are asymptomatic at the time. Quarantine helps prevent the potential spread of the disease if these individuals have symptoms later. All right, so now how can screening and contact tracing be used to address the spread of communicable disease? So ideally, the same criteria used for screening non-communicable diseases can be applied to communicable diseases, which are the disease produces substantial death and or disability. Early detection is possible and improves outcome. There is a feasible testing strategy for screening, and the screening is acceptable in terms of harms, cost, and patient acceptance. And the screening process involves detecting diseases early and identifying those at risk to reduce further transmission. Then we also have contact tracing, which identifies, monitors, and supports individuals in close contact with a diagnosed or probable case of disease. Contact tracing has been traditionally a key role of local health departments, and it has been particularly effective in controlling diseases like tuberculosis and sexually transmitted diseases. It's also a critical part of screening for asymptomatic diseases and case finding, which are clinical diagnosis in the presence of symptoms. Now, there are some conditions that do make contact tracing effective. First up, we have a long serial interval. Contact tracing works best when there's a long enough time period between exposure and the development of symptoms, allowing enough time to identify and track contacts. Then there also needs to be a clear-cut route of transmission. For contact tracing to be effective, there needs to be a clear and identifiable route through which the disease is transmitted, like sexual contact or droplet transmission. We also need effective treatments, which allow health authorities to manage and treat those identified during contact tracing. And also if the disease is relatively rare, because contact tracing is especially useful for relatively rare diseases that might not have widespread exposure, but where identifying each contact is crucial to preventing further spread, as opposed to COVID, where everyone was getting it at one point. Right, so now how can immunizations be used to address the burden of communicable disease? Well, vaccinations stimulate the development of active immunity, meaning the body's own immune system is triggered to recognize and fight off specific pathogens, and offers long-term protection. And traditional vaccines generally fall into two categories. First off, we have the inactivated vaccines, or dead vaccines, where dead organisms are injected to prompt the immune response. Then we also have attenuated or live vaccine, where weakened live organisms are used, providing long-term immunity by stimulating cell-mediated immunity, and both types help the body produce antibodies to fight off the disease. Now, next up we have what we call our next generation vaccines, which are a more recent development, with mRNA vaccines and viral vector vaccines being the key examples, especially for COVID-19. Both these deliver RNA to the cells that allow them to produce the COVID-19 spike protein. However, there is limited experience in the use of these vaccines in humans, so it is very important to thoroughly investigate their safety before widespread use. mRNA vaccines work by delivering the virus's genetic RNA into the recipient's cells. These cells then produce the protein coded by the RNA, like the COVID-19 spike protein, and the immune system responds by producing antibodies and cell-mediated immunity. The next are for viral vector vaccines. They use a harmless virus, which doesn't reproduce in humans, to deliver the RNA, allowing cells to produce this viral protein. This method typically requires more virus particles to deliver the RNA, but it has some advantages over the mRNA vaccines, in that they can be stored at refrigerator temperatures, making them cheaper to produce and to distribute them than our mRNA vaccines. While not as effective at preventing disease overall, they're still around 80 to 90% effective at preventing severe disease, they still provide strong protection. Both mRNA and viral vector vaccines have a limited duration of effectiveness, meaning boosters are required to maintain immunity. Now, some safety considerations that all vaccines, including the mRNA and viral vector vaccines, can produce side effects. All right, so now how can antibiotic or antiviral treatments help address the burden of communicable disease? Effective treatment can also act as a form of prevention because it reduces the likelihood of transmission. So for example, in HIV treatment, antiretroviral drugs help reduce the viral load in the patient, making it much less likely for the virus to be transmitted to others. And this approach known as what we call the epidemiological treatment, or contact treatment. This involves treating individuals who may have been exposed to the disease, even if they're not showing symptoms yet. And the reasoning behind it is that early stage or low-level infections might be difficult to detect, but treating these contacts early on can significantly reduce the spread of the disease.